Chapter 17 is our last chapter of the semester. So this is a chapter that talks about nucleic acids and DNA and RNA. And before we talk about those, we're going to start off by talking about nucleosides and nucleotides, which are like the building blocks for those molecules. So it's very similar to with proteins, how amino acids are the building blocks of proteins. For DNA and RNA, which are both nucleic acids, nucleotides are the building blocks for those. So just in terms of an introduction here, nucleic acids are going to be polymers that are not branched. So remember, branching is whenever we looked at sugars and we had some molecules like glycogen, for instance, which were polymers of sugar, but there was branching. It kind of went off in all kinds of different directions. For nucleic acid, they're unbranched, right? So unbranched meaning that there are going to basically be linear chains of these repeating units, and the repeating units are going to be nucleotides. And there are two main types of nucleic acids that we're going to look at. There's going to be DNA and RNA. So DNA is deoxyribonucleic acid. That's the stuff in you that stores your genetic information, and that you can also transmit it whenever you have a kid, for instance. You transmit some of your genetic information to your offspring. RNA, or ribonucleic acid, takes that information that's in your DNA and translates it into proteins that can be used, like we talked about in the previous chapter, things like enzymes and all the other things that proteins can do. So RNA helps do that translation. There's actually different types of RNA, and most of them are involved in that translation of DNA into proteins in some shape or form. Nucleotides have three parts to them. So nucleotides have a nitrogen-containing base, and there are 5 different bases. And we'll look at the different structures of those 5 bases coming up here soon. Then you have a monosaccharide, so you have a sugar component down here, and then you're going to have a phosphate. Nucleotides can either have 1 phosphate, 2 phosphates, or 3 phosphates. So they can actually have multiple phosphates attached to it. And we'll look at different examples of those coming up here in just a few minutes. So again, a nucleotide is going to have a base, a sugar, and a phosphate as its main 3 parts. Now, DNA molecules are going to have millions of nucleotides linked together. You can actually have shorter DNA molecules, but the DNA molecules, for instance, that make up your chromosomes, which contain the genetic information of your body, are typically in the millions of nucleotides long for each chromosome. And if you were to look at the total amount of DNA in your body, the number, you would actually have about 3 billion or so nucleotides in your body that make up your genetic information. So 3 billion different nucleotides, that the sequence of them basically leads to us being who we are as humans. Those 3 billion nucleotides comprise 23 pairs of chromosomes. So if you've heard of the company, say 23andMe, this is why it's 23, right? Because there's 23 pairs of chromosomes. And each of those chromosomes are made up of many different genes. So a gene is going to be the part of that DNA molecule that makes a protein. So you imagine if you have a very long piece of DNA, let's pretend that's our DNA. That region there might be a gene, that region there might be a gene, that region there might be a gene. Now you have other regions, right, that those arrows are pointing at, that don't make genes, but there are still important pieces of DNA in your genome which help regulate that gene expression. We used to just call that like junk DNA, and we used to think that it really wasn't important anything. But as science has uncovered more and more of information in terms of what all of the base pairs do within our genome, we've actually shown that a lot of this DNA that used to be considered junk DNA because it didn't make a particular gene was very important for controlling how those genes are expressed into proteins. All right, so before we actually talk more about the nucleotides, we're going to talk about nucleosides. So nucleosides with an S here. Nucleosides is when you have a sugar and a base together. So sugar plus base. All right, so that's going to be a nucleoside. So in RNA molecules, your sugar is going to be ribose. So when I say in RNA molecules, so in other words, if I were to look at these structures down here, this one and this one, I can say that the one on the left, which is the sugar ribose, that that sugar itself is going to be found in RNA. Whereas the one on the right that says deoxyribose, see where it says deoxyribose there, that's going to be one that's present in DNA. And the only difference between the two is if you look at the name of it that says D and then 2-deoxyribose. So thinking about what you know about naming organic molecules, you can imagine if we were to number our carbons in this 1 through 5, you can see the numbers over here. So this is going to be carbon 1, 2, 3, 4, and 5. 2-deoxy means on carbon 2, there is no oxygen, right? Whereas over here in regular ribose, on carbon 2, there is. So that's where deoxyribose, the meaning of the name, comes in. That deoxyribose is going to be what is present in DNA. Right? So for both of these, the sugar is going to be ribose or deoxyribose. Now, we said for a nucleoside, it's a sugar attached to a base. So we just talked about the sugar. Now, the base is going to be one of 5 different bases. And you don't need to memorize the structures of these. But what I do want you to know is whether the base is a pyrimidine, which is what we're looking at on this slide, or the next slide is going to look at purines. So the bases can be lumped into one of 2 categories, pyrimidines or purines. So pyrimidines kind of look like this compound over here. And the main thing I want you to see about that is it has 1 ring. So it's a ring that has nitrogens in it, so it has 2 nitrogens, but it's a single ring. So Cu and T are cytosine, uracil, and thymine. So these are the bases that are all pyrimidines, and they have 1 ring. All right. Now we compare that to purines on this slide. Notice that purines have 2 rings. So purines are going to be A and G. All right, so purines, 2 rings are going to be A and G, and pyrimidines have 1 ring and they're CUT. All right, so whenever we look at our nucleic acids, so DNA and RNA, there's slight differences between the bases that are going to be found in those. So DNA is going to have A, G, C's, and T's. So adenine, guanine, cytosine, and thymine, whereas RNA is going to have A, G, C, and U. So in DNA, you have a thymine, and in RNA, you have uracil. So I'm going to go back to the previous slide here, and basically, so this one here is uracil. This is the one that's going to be found in RNA, and thymine over here is going to be found in DNA. So there's an extra methyl group here on the one that's found in DNA. Okay, now that we've talked about a nucleoside in terms of the 2 parts, the sugar and the base, now let's look at how we kind of link those together. So nucleoside is formed by joining the carbon of the monosaccharide, specifically carbon 1 of the monosaccharide, with a nitrogen from the base. And then whenever we name the nucleosides, they're going to end in idine or ocene. And I'm not too worried about you actually being able to name your nucleosides. So don't worry about that too much. Apart from that, I do want you to understand this prefix of deoxy and what it means. So if you hear deoxy, you should know that that's referring to that oxygen on carbon 2, and you should be able to pick that out of a structure. All right, so how do these nucleosides get formed? Right, so carbon 1 is right there, carbon 2, carbon 3, carbon 4, and carbon 5 on D-ribose. So the carbon on carbon 1 is going to link with one of the nitrogens on your base, and what you form, looking over here on the right, it's called an N-glycoside. So basically, glycoside refers to the linkage between a sugar, and N-glycoside means that that sugar is linked through a nitrogen, right? Just like we would use the prefix back in naming like amines or amides, we would say like N-dimethyl, whatever, N-dimethyl benzamide, right? And we would say that those methyl groups would be attached to a nitrogen. In this case, it's saying that the glycoside, the sugar, is attached to the nitrogen, right? So cytidine would be this one here. And again, I wouldn't expect you to pull out of a lineup cytidine. Apart from, you should be able to know if I showed you 4 structures, like say for a multiple choice question, and only one of them had a single base and the other ones had 2 bases up there, right? You should be able to pick out cytidine from that, because you would know that cytidine is a pyrimidine, whereas anything with 2 rings would be a purine. Okay, so here is same example as before. This one is just showing you something with a deoxyribose, and this one, notice there's no OH on carbon 2, and you form this N-glycoside. Okay, so that talks about nucleosides. So now we're going to make the leap to nucleotides. So nucleotides are going to be the same thing as a nucleoside, except that on carbon 5, right here, we are going to add a phosphate to it. So again, looking at cytidine here, we're going to add a phosphate to it, and that's going to make a nucleotide. So the resulting nucleotide, right, if you look at the name down here, it says it's cytidine 5' monophosphate. If you're wondering what 5' means, in terms of numbering the carbons in the base get numbered 1, 2, 3, and 4. And we rarely talk about those carbons in their numbering. But because they get numbers 1, 2, 3, and 4, in the sugar we number them 1', 2', 3', 4', and 5'. So that prime designation there basically refers to the fact that it's on the sugar. So a 5' phosphate means that on that carbon number 5, where there used to be an OH, we're going to replace that OH with a phosphate, with this PO4 group. One thing to make note of with the nucleotides, right, that phosphate group is negatively charged. Negative charge. That's going to be important whenever we look at kind of the structure and properties of DNA, right? So the nucleotides that make that, that are part of that, the nucleotides that are in DNA are going to have this phosphate to it, and they're going to have a negative charge because of that. All right, last thing on this slide, talking about the name. So this, again, says cytidine 5' monophosphate. So whenever we name nucleotides, we name them with abbreviations. And what happens is the base, so cytidine, turns into a C. So that first letter can either be, whoops, go back here, A, C, G, T, or U, right? Because those are the 5 bases, all right? The next letter, where it says M, stands for mono. So that next letter can either be M for mono, or we can have, we can also have D for di or T for tri. So di or tri. So you can have 1 for mono, 2 for di, 3 for tri, phosphates. So that second letter is either going to be M, D, or T. Then the last letter is always going to be P. So in other words, you can have CMP, like is shown here. You could also have CDP or CTP. You could have AMP, ADP, ATP, and so on and so forth for all of your other nucleotides. Now, there's actually one little additional piece of information to that, which is shown on the next slide here. So again, here we are showing that you have the phosphate being added to the deoxyadenosine. So this one because it's a deoxyribose, or deoxyribose sugar is part of the nucleotide it's going to form, we're actually going to give it a different prefix. So it also gets a lowercase d in front of it. So that lowercase d tells you that you have deoxyadenosine 5' monophosphate. So D stands for your deoxy, and then adenosine monophosphate. And in the structure down here in the bottom, it mentions 5' monophosphate. There are times where this phosphate can be linked to other places, particularly this 3' OH. We're not going to worry about that in this class, though. So as far as you guys are concerned, no, that phosphate is always going to be attached to the 5' carbons. That's what I want you to know. Phosphate goes on the 5' carbon. Here's an example of a diphosphate, so ADP. Notice this time there's no lowercase d in front of the A, and that's because you do have an OH here on carbon 2. And this one again shows you have two phosphates linked together here. So this would be ADP. And then finally this one here is ATP, where you have three phosphates linked together. And again, I wouldn't expect you, if I were to show you this structure on an exam, I wouldn't expect you to be able to name it as ATP necessarily. What I would be able to expect you to pull out is that, one, there are three phosphates, so you should know that it's going to be something TP. You should be able to notice that there's an OH there, so, right, it can either be D-ATP or regular ATP. So whenever you see the OH, that means it's going to be a regular something TP. And then based on the fact that there are two rings here, you should be able to tell me it's either going to be ATP or the other option that it could be would be GTP, right? Those could, because those are the two that are purines. So in other words, if I did show you this structure on a test and I said, what's the name of it? You should be able to pick it out if the multiple choice options were ATP, and then another one was maybe going to be GDP, because that's wrong, because you know that the D is wrong, right? And if another answer was, say, UTP, well, you would know that's wrong because you know that a U would only have one ring because it's a pyrimidine. And let's say my other option was DATP. You would know that one is wrong because the D would mean that there's no oxygen there, so that has to be wrong. So for a multiple choice question, you should still be able to answer it and come up with ATP as an answer. All right, so in terms of doing some problems like this and naming the molecules, right, so what can we say about this first one? So the first one, we can look at it, whoops, and say that this is going to be a nucleoside, right? So by just looking at it, we can say it's going to be a nucleoside. And I know that because there's a base, sugar, and there is not, so no, phosphate. So the P with the circle is going to be my abbreviation for a phosphate. So this one has just a base and a sugar. I can furthermore say that this one is going to be a purine, right, because it has the two rings. So we know that it's either going to be adenosine or guanosine based on that. And we can also look at it and say, oh, it has an OH there, right? So we know that it's not a deoxy. So this is one that would go in RNA. So, right, so this would be either adenosine or guanosine, right, based on what we know. And it's actually going to be the first one. It's actually going to be adenosine here. But again, I wouldn't expect you to be able to distinguish between the two. Now, if you're looking at the bottom one, right, so the bottom one, I can say it's a nucleotide, right? And how do I know it's a nucleotide? Because it has a base here, it has a sugar here, and then it has a phosphate over here. So it has all three parts. So it's going to have base, sugar, and a phosphate. Okay, so it's going to have all three. We can say that in terms of the base, we know that this one's going to be a pyrimidine. a pyrimidine, right? So this one is going to be either C, U, or T, all right? And then the last thing we can say is that there is no OH there. So if I were to tell you that this one actually was a C, right? You could name this one, and you could say it's going to be deoxy, right? Because there's no oxygen here on carbon 2, so we could put a lowercase d. Then we would have C, and then because there's only one phosphate over here, we could say it's going to be monophosphate, and it would be DCMP would be your answer for what that is.